Imagine a world where organisms can't run or walk, yet they've somehow mastered flight, swimming, even explosive jumping to colonize, well, pretty much every corner of the planet.
Sounds like science fiction, doesn't it?
It really does. But these are fungi, and today we're taking a deep dive into their fascinating, often invisible universe of spores. You might think you know fungi, but really, their incredible success, their ability to be almost anywhere, it all comes down to these tiny, specialized biological units.
It really does.
Our mission today is to unpack the ingenious ways fungi disperse these spores, what their sheer numbers mean for us, and the surprising impacts they have on our world and actually our health.
That's right. And the truly remarkable thing is just how vital these microscopic structures are. And the abundance is just mind-boggling.
How much?
Well, the air we breathe right now,
Ah.
it can contain over 10,000 spores per cubic meter.
Wow.
So yeah, we'll explore the why behind those numbers, the how of their sort of intricate dispersal mechanisms, and the significance they hold for you. Hopefully it gives you a completely new perspective on the air around you.
So get ready to see the invisible world around you in a whole new light. We're going to walk you step by step through how these amazing little units navigate the world and how they impact everything from plant diseases to your own well-being, often without you ever seeing them. Let's start with that fundamental question then. Why do fungi produce so many spores?
Right.
They're master colonizer, sure, but their secret weapon seems to be just incredible numbers. They create countless tiny specialized spores, ensuring that wherever a new food source pops up, they're there, ready to go.
Exactly. So the obvious question then is, why this sheer abundance? I mean, fungi don't have legs or wings, right?
Right.
They can't actively hunt for new places to grow, so they have to rely on overwhelming odds. It's a numbers game.
Here's statistics.
Pure statistics. The more spores released, the higher the chance a few will land in just the right spot to grow into a new mycelium, the main body of the fungus.
And we're talking big numbers here.
Oh, huge. Some common fungi release billions of spores daily for months on end.
Billions.
Daily.
Yep. Take the common bracket fungus, Ganoderma applanatum. A single one can discharge an astounding 30 billion spores a day.
30 billion.
Which adds up to maybe 4.5 trillion spores over a single season.
That's astronomical.
It is. Even a tiny penicillium colony, maybe an inch across, it can churn out 400 million spores. So this sheer volume means that even if they're spread super thin across the atmosphere, there's always a significant number of spores around us just waiting.
Okay, that is an astonishing amount. So with that many spores needing to get around, how exactly do they do it? Without legs or wings, what are some of the most surprising transport solutions evolution has cooked up?
It's truly a marvel of natural engineering. Fungi have evolved this just bewildering array of strategies, often inventing similar solutions completely independently, like convergent evolution.
Exactly. We can broadly categorize these by their primary mechanism. Let's start with the aquatic ones.
Okay.
For fungi living in water, like the chytridiomycota and oomycota, the solution is pretty simple. Swim.
They swim.
Yeah, their spores are called zoospores, and they're equipped with flagella, those little whip-like tails for propulsion through water.
Little swimmers.
Little swimmers. And what's surprising is that even among these tiny guys, there are distinct styles. Some have one backward-pointing tail, kind of like a sperm cell. Others have forward-pointing flagella, sometimes with little hair-like bits for extra thrust.
It's not just random swimming, though, is it? They aren't just bumping around hoping to find food.
No, exactly.
Right.
That's the really cool part. Many of these aquatic spores have a special talent called chemotaxis.
Chemotaxis. Okay.
Which is the ability to navigate towards a food source by sensing a chemical gradient, like following a scent trail.
So they can smell their food.
In a way, yeah. For instance, Pythium and Phytophthora zoospores, which can be nasty plant pathogens. They can trace sugars and other chemicals leaking from plant roots to find their hosts.
Wow.
And some chytrids that attack nematodes. They can detect and swim towards substances coming from the worm's orifices. So it isn't just movement, it's guided navigation. A microscopic homing beacon to their next meal.
Guided navigation. That's definitely efficient in water. But what about fungi that need to spread over land? Or from one plant leaf to another? Swimming just won't cut it then.
No, absolutely not. That's a powerful point about selection pressure, right? When swimming wasn't an option, fungi evolved two completely new structures that really changed biological history.
Okay, what were they?
The aerial sporangiophore, basically a stalk lifting the spore factory up into the air, and the detachable wind-dispersed mitosporangium, which is the spore-containing sac itself.
Up into the wind.
Exactly. We see this with downy mildews, which are actually oomycetes, related to the swimmers. Their microscopic sporangia develop on these slender branches, very easily dislodged by wind or rain.
Okay.
Now, when they land on a new leaf, most still need a film of water to release their swimming zoospores. But some highly evolved species, like in the Paranosbraceae family, they can directly produce a germ tube. They've cut their last link to needing that aquatic life phase.
They've become fully terrestrial, basically.
Pretty much, yeah. At least for that stage.
The wind is a powerful ally, obviously, but sometimes the most effective dispersal agent is, well, us.
Oh, absolutely. What happens when humans unintentionally become a fungus's fastest way to conquer a continent?
Like the potato blight.
Exactly. The oomycete Phytophthora infestans, the cause of late blight of potato. Human transoceanic commerce inadvertently carried this fungus from Central America to North America in 1843, then boom, across the Atlantic to Europe in 1845.
And caused the Irish Potato Famine.
Devastating consequences. It's
Huh.
just a powerful testament to how quickly a tiny spore can travel the globe with a little unintended help. Changed history.
Absolutely, a truly global traveler indeed. Okay, so what about other groups? You mentioned zygomycetes.
Right, let's turn to the zygomycetes and their asexual stages, what scientists call anamorphs.
Anamorphs, so they're non-sexual forms.
Exactly, the forms they use just for quick reproduction and dispersal. And they show an incredible range of strategies.
We see such ingenuity here. Some zygomyces produce large sporangia, these big sacs filled with hundreds or thousands of spores. In species like mucor, these spores are embedded in this sticky slime, like a spore drop, designed for dispersal by small animals brushing past.
Right, hitching a ride.
But in others, like Rhizopus stolonifer, the common bread mold, the spores are dry. They just blow away on the wind when their protective sac breaks open.
Two very different approaches right there.
Yeah. And then there's the really dramatic stuff, violent discharge.
Ah, Pilobulus, my favorite.
It's amazing. This ingenious technique seems unique to this one genus, Pilobulus, which lives in herbivore dung.
Right, not the most glamorous address.
Maybe not. But to survive, it has to get its spores away from the dung and onto the surrounding grass where grazing animals will eat them.
So it needs some serious range.
It does. This fungus has a unique structure, a subsporangial vesicle that builds up internal pressure, like serious pressure.
How much pressure are we talking?
It ruptures at about 7 atmospheres.
7 atmospheres. That's roughly 7 times the pressure in your car tires.
Exactly. It forcefully expels its spore mass, plus a bit of glue, up to 2 meters away.
2 meters from a microscopic fungus.
Yeah, far enough to clear even a big pile of dung, getting the spores onto the animal's next meal.
That is basically a microscopic pressure cannon.
Wow.
I guess you wouldn't want to be standing too close when that thing goes off.
Probably not.
What this incredible variety within the zygomycetes really highlights is just nature's relentless pursuit of efficient dispersal. Combining different approaches, sticky, windy, explosive, sometimes even within closely related species.
So we've seen explosive launches with Pilobulus, a truly dramatic method, but not all fungi need such high-octane solutions, right? What about those that use maybe a more refined approach, or even a dual strategy?
Indeed. Some fungi hedge their bets. They produce both large, slimy sporangia for animal dispersal and small, wind-dispersed ones at the same time, allocating resources in 2 different ways.
Covering all bases.
Exactly. Others specialize in what look like beads on a string. Those are called merosporangia, or even just single-spored sporangia.
They're like the ultimate in downsizing.
Right. And these can be dry and wind-dispersed, like in Cunningamella, or slimy and sticky for animal hitchhikers, like in Kixella. Some dung fungi, like Spirodactylon, even have these tall branched structures, specifically designed to tangle in rodent hair.
Ensuring ingestion during grooming, that's devious.
It's effective. And for even more active projection, we have the entomophthorales. They're one-spored mitosporangia, are basically ballistospores.
Ballistospores, meaning they're shot.
Actively shot away, yeah. By different clever mechanisms, often involving a burst of pressure or the rupturing of a specific part of the structure to propel them into the air.
Okay, now let's talk about the big guns. Or maybe, in some cases, the surprising lack thereof. Ascomycetes and Basidiomycetes. These are the 2 largest fungal groups, right?
They are. And though they look very different, they both evolved really sophisticated spore-shooting mechanisms.
Which are key to their success.
Absolutely. For Ascomyces, the ascus, which is this microscopic tubular cell, it seems to have originally evolved as a true spore gun.
A spore gun. Okay.
Turker pressure builds up inside the cell until the tip bursts, launching the ascospores.
Yeah.
And fungi developed ingenious ways to build this pressure. Some asci are unituticate single wall. They might pop open like a little lid where the tip stretches elastically.
Okay.
Others are bitunicate. They have this clever double-walled structure where the inner wall balloons outwards, pushing the spores out with a sort of secondary burst. Different designs, same powerful result. Shoot the spores.
Imagine hundreds of thousands of these tiny spore guns firing all at once.
You can actually see it. That's what happens in puffing apothecial ascomata. Those are the cup-shaped fruiting bodies. When humidity changes, poof, you see this smoke-like cloud released into the air.
Wow.
This collective blast generates its own air movement, carrying the spores much farther than if they fired individually. For example, Cucanus sulcipes, a really colorful tropical cup fungus, produces an astonishing 1.7 million ascospores per square centimeter.
Incredible density.
And some even aim their spores towards the light, showing a level of sophistication you might not expect.
Aiming? How?
Phototropism. They grow toward the light source before firing. And the size of the projectile matters too, right? Just like with a cannon.
Makes sense.
In the dung-inhabiting Saccobolus, the 8 ripe ascospores are actually glued together. They get expelled as one single larger projectile. More mass, more momentum travels farther.
Smart.
Apodospora fimicola, which has exceptionally large ascospores, can shoot them a phenomenal 50 centimeters.
Half a meter for a single cell structure.
It's amazing.
But not all archaea shoot their spores, do they? What happens when that shooting mechanism just isn't useful anymore?
You're right, evolution is practical. That shooting mechanism is often lost when it's no longer needed. Like in fungi that fruit under tree bark or underground. Like truffles.
Wow, truffles.
These fungi have spherical ascae, all contained within closed fruiting bodies. They rely entirely on mammals for dispersal.
Like pigs finding them.
Exactly. Female pigs were traditionally used to find truffles because they're attracted by aromas that actually mimic a boar pheromone.
No way.
It's a bizarre but highly effective chemical dispersal strategy. Similarly, Ophiostoma, the fungus that causes Dutch elm disease, relies on bark beetles. It oozes out a sticky spore drop that the beetles pick up and spread.
And that spread can be incredibly fast.
Devastatingly fast. Its rapid spread across North America after humans accidentally introduced it in the 1930s just highlights the power of these vector-borne strategies.
Okay, prepare us for the grand finale of fungal ingenuity here. I heard some fungi perform a full-blown acrobatic show just to launch their spores. Tell us about Phylactinia.
Oh, Phylactinia. You won't believe this one. This powdery mildew starts with what looks like a tiny spiky ball on a leaf.
Okay.
Those spokes aren't just decoration. They actually act as livers, prying the entire spore packet, the whole ascoma, right off the leaf. So now it's airborne.
It launches itself.
It levers itself off.
Yeah.
But it's not done. It then lands, usually upside down, sticks to a new surface with some mucilage, and then almost like a tiny self-opening clam, it splits right around its equator.
It splits in half.
The bottom half swings open 180 degrees, exposing the asci inside, which finally get to point outwards and shoot their spores.
That's insane. A multi-stage, almost theatrical dispersal system.
Absolutely. And this incredibly complex dance helped this fungus spread devastatingly across Europe in the mid-1800s, crippling grape crops, all thanks to these spores getting around.
Wow. Okay, now to the basidiomycetes. Specifically, their basidiospores. You said these are typically projected much shorter distances, usually less than a centimeter.
That's right. Much shorter range than the ascospores.
Why the difference?
Well, if we connect this to the bigger picture, it's all about adaptation to their specific fruiting body.
Yeah.
The mushroom.
Okay.
A mushroom's gills represent this huge spore-producing area, right? But they're very, very closely packed together.
Right, very thin spaces between them.
Exactly. If a basidiospore were shot too far, it would just smack into the next gill. Game over.
Ah, okay.
So the evolution of agaric's gilled mushrooms has fine-tuned this really delicate shooting mechanism. Spores are launched just far enough to clear the gill they came from and fall straight down into the airspace between the gills.
And then the wind takes over.
Then they get carried away by air turbulence below the cap. This is why most mushroom caps are shaped like a biological umbrella.
To keep the spores dry.
Exactly. To protect the hymenia, that's the spore-producing surface, from rain. If those delicate gills get wet, the surface tension mechanism that launches the spores gets disrupted, and they stop shooting.
That umbrella shape makes so much sense now. And some mushrooms have an even more a dramatic way of ensuring their spores get out, don't they? Like they
Yeah.
melt.
That's autolysis, self-digestion. What's fascinating here are strategies like Coprinus comatus, the shaggy mane mushroom.
I think I've seen those.
Probably. Its cap is deep and cylindrical, with extremely thin, tightly packed gills. Spores are released in this highly regimented wave, starting right from the bottom edge of the gills.
Okay.
And this spore release is followed immediately by a wave of autolysis that literally melts away the gill tissue that just released its spores.
It digests itself.
It digests itself,
Sure.
continuously exposing new baidia, the spore-producing cells higher up, allowing them to shoot their spores in turn. You can literally watch the entire cylinder of gills gradually melt away into black goo as the spore shooting progresses from bottom to top.
That's an elegant, if slightly gruesome, self-sacrificing system.
It really is. Maximizes dispersal.
It's incredible how specific these adaptations are. You mentioned you can even tell if a basidium is actively shooting its spores just by looking at it.
You can.
Yeah.
Under a microscope, of course. If the spores are mounted asymmetrically on their little stalks, the sterigmata, they're actively propelled, like they're offset, ready to launch.
Okay.
But if you see symmetrical mounting, that means no active propulsion. It signals a radical evolutionary change has happened.
What kind of change?
That change often leads to what we call sequestrate forms. These are evolutionary offshoots that have lost the ability to shoot spores. They often have crumpled gills or entirely enclosed spore-bearing tissues.
So they need another way out.
Exactly. Many are hypogous, meaning they fruit underground. Think truffles again, but the mycete versions. This adaptation likely arose in response to dry conditions, making them reliant on insects or mammals for dispersal, like the California redback vole that almost exclusively eats rhizopogon truffles and then, well, distributes their spores later.
Nature finds a way. And then there are the gastromycetes. That's a wild group, right? Puffballs, earth stars, bird's nest fungi, stinkhorns.
Oh yeah, the gastromyces. They all have specialized, often bizarre dispersal methods because they've generally lost that active spore shooting.
So how do puffballs work?
Puffballs release a mass of dry spores.
Mm-hmm.
That mass is called the gleba, through a hole in their papery shell. When raindrops hit the outer layer, it creates a little puff of air, forcing spores out.
Like a little bellows.
Exactly. Earth stars do something similar, but they first lift their spore sac up on these star-like rays, getting it above the leaf litter. Bird's nest fungi have their spores packaged into tiny, seed-like packets called peridioles.
The eggs in the nest.
Right. They sit inside a splash cup structure. Falling raindrops hit the cup and splash these eggs out, sometimes quite a distance. And then there's the tiny earth ball Spherobolus dilatus, only 2 millimeters wide, but it can literally catapult its entire spore mass up to 7 meters.
7 meters.
By an explosive, pressure-driven inversion of its inner cup, like turning itself inside out very, very quickly.
Incredible power in something so small. And the ultimate showstopper might be the stinkhorns, right?
The stinkhorns, yes. Their young fruit body starts as this thing called an egg. Then it rapidly elongates into a tall, spongy stalk. And the top is coated with this sugary but absolutely foul-smelling greenish slime embedded with the basidiospores.
Evil smelling is right.
Oh, it's potent. But that smell is irresistible to certain flying insects like flies. They flock to it, gorge on the sugary slime.
And carry the spores away on their feet.
Exactly. Mission accomplished. Some, like acero, even add a visual lure, bright red, radiating rays like some bizarre flower. The combination basically screams flounder, rotting meat, or feces to a passing insect.
Truly strange fungi indeed, combining flowers and, well, excrement for dispersal.
Nature isn't shy about using what works.
Okay, so all these incredible, sometimes weird dispersal mechanisms lead to one undeniable fact. Spores are absolutely everywhere.
Everywhere.
And while they're vital for fungal survival, their omnipresence, well, it can have significant implications for us.
Right. So the obvious question then becomes, what significance do these astronomical numbers hold for us? Remember, spores are microscopic, self-contained units. They have everything needed to germinate. And they're released pretty much constantly from early spring to late fall, and even in winter if it warms up.
For a long time, respiratory allergies, hay fever, were mostly blamed on plant pollen.
That was the main suspect.
But scientists soon realized that many allergies persist right through fall and winter, long after most pollen is gone.
Exactly. That's when fungal spores became prime suspects. Skin tests confirmed it. They are definitely allergenic.
How many people are affected?
It's estimated about 20% of the population is apopic. That means they're easily sensitized by just the normal, everyday concentrations of spores in the air. That leads to what people commonly call hay fever symptoms, or it can trigger asthma.
And the other 80%.
For the other 80%, it generally takes higher concentrations to cause problems, like the levels you might encounter during haymaking or grain handling.
Okay, so occupational exposure.
Often, yes. These higher concentrations can lead to conditions like allergic alveolitis, also known as hypersensitivity pneumonitis. It causes inflammation deep in the lungs, leading to breathlessness, often affects farmers or grain handlers.
Is it related to farmer's lung?
It is. Farmer's lung is one of the most serious examples. It's a potentially fatal allergic disease caused by repeated exposure to really high concentrations of spores from moldy hay.
How does it manifest?
Well, there's often an acute stage seen in harvesters: chills, fever, cough after brief but overwhelming exposure. But maybe more dangerous is the chronic stage, often seen in silo workers who get constant low-level exposure.
Mm.
That can lead to degenerative respiratory changes, airway obstruction, even emphysema. It was first described way back in 1924 in Canada, and it's pretty common in temperate farming regions.
And it's not just farms, right? Indoor environments, too.
Oh, absolutely. Damp buildings, poorly maintained air conditioning systems, they can become major sources of allergenic spores. We see similar respiratory complaints in office workers sometimes, trace back to the building's air system.
Okay, here's where things get maybe a bit surprising and definitely controversial. Back in 1993, there was this black mold, Stachybotrys chartarum, grows on damp wallboard.
Right, the toxic black mold.
It was tentatively linked by the Centers for Disease Control, the CDC, to a serious condition called bleeding lung syndrome in infants in Cleveland.
I remember that.
And this led to some really serious actions libraries being closed, proposals to demolish and rebuild entire hospitals costing millions and millions of dollars.
Huge costs, yeah. So the big question was, were those administrative decisions reasonable, based on the science?
What did the science say?
Well, while the CDC did record many instances associating health problems with Stachybotrys, they later had to admit there was no solid proof of causation, only correlation.
Ah, the classic correlation versus causation issue.
Exactly. As scientists always stress, correlation does not equal causation. And jumping to conclusions based only on correlation can be extremely costly, as we saw there.
So some scientists think Stachybotrys kind of became a scapegoat.
That's a view held by quite a few, yeah. While it definitely produces some nasty mycotoxins, those toxins have really only been proven to cause problems in animals when they're ingested, like horses eating large amounts of moldy hay.
But people aren't usually eating their walls.
Generally not, no. In buildings, Stachybotrys usually grows inside wall cavities or in crawl spaces, not typically where humans are eating it.
So the risk isn't ingestion. What about breathing it in?
That's the key point. And critically, Stachybotrys spores are produced in these tiny, slimy droplets. They're sticky. They generally stay put and aren't easily released into the air.
Unless
the colony dries out completely and the spores are physically disturbed, which often happens, ironically, during active removal attempts if not done properly.
so disturbing it can make it worse.
It can temporarily increase airborne spores, yes. So the scientific consensus, as of around 2017 anyway, tends to be, don't panic. The focus should be on preventing the water intrusion in the first place, fixing the leak, drying things out, and using simple cleaning agents like bleach on surfaces. Not necessarily resorting to extremely expensive, drastic measures based on unproven causation. Address the moisture. That's the best remediation.
Okay, that's a really important clarification. So given the impact airborne spores clearly do have on plant diseases, lung infections, allergies, it seems crucial that we can actually quantify and identify them.
Absolutely essential.
But with nearly 100,000 known fungal species out there and many spores looking frustratingly similar, that sounds incredibly difficult.
It definitely presents challenges, but fortunately the situation isn't quite as dire as it might seem. Why? Because the vast majority of spores floating around in the air actually come from a relatively small number of common fungal genera.
Ah, okay, so it's not evenly distributed.
Not at all. Researchers like Grant Smith and others have developed methods specifically to identify the most common allergenic spores. We generally use two main approaches.
What are they?
First, there are viable culturing methods. This involves trapping spores on a nutritive agar medium, like a petri dish, with food, and then waiting for them to germinate, grow into a little colony, and hopefully produce more spores.
So you can see what grows.
Exactly. This allows for pretty good species-level identification for some common molds like Aspergillus and Penicillium.
But what are the downsides?
Well, the big downside is that many important spores, like those from rust fungi or most mushrooms, simply won't grow on standard lab media. And this method completely ignores any dead spores, which might still be allergenic.
Okay, so it gives you part of the picture, but not all of it. What's the other method?
The second approach is non-viable observational methods. This traps spores by impaction, basically slamming them into a surface, often onto a greased microscope slide. Then you just look at them directly under the microscope.
So you count what lands there.
Right. This method detects a much wider range of fungal types, including all those spores that won't grow in culture. And importantly, it counts both live and dead spores.
Yeah.
So it's often recommended for general air quality surveys because it gives a much broader, more representative picture of what's truly in the air.
You mentioned impaction traps.
Yeah, devices like the sampler impaction trap, for instance. It uses a small fan to suck air through a narrow slit, and the spores in that air get slammed onto a grease slide that slowly moves past the slit. This collects sequential samples over time.
So you can see how spore levels change hour by hour.
Exactly. It gives scientists a real-time or near real-time snapshot of the air quality and what's floating around.
What have these global studies actually revealed? Who are the main culprits in the air?
It's fascinating data. A big meta-analysis looking at over 200 reports from around the world showed that one genus, Cladosporium, mostly the species C. herbarum, represents, on average, about 33% of all airborne fungal spores.
A third. Just one type.
Roughly, yeah. Then Basidiomyces, mostly mushroom spores, make up about 22%. Ascospores, around 14%. Alternaria, another common mold, about 4.5%. And Aspergillus penicillium types together about 3.5%. Many others are present, of course, but those are consistently the most abundant players globally.
And these counts aren't static, right? They change.
Oh, definitely not static. They vary hugely. Seasonally, for instance, you see mushroom spores peak in the fall in temperate zones, while certain rust spores might peak in spring and then again in fall, depending on their life cycle.
And even day to day.
Even hour to hour.
Huh.
There are daily fluctuations. Cladosporium counts often peak around midday, probably due to higher wind speeds and drier conditions releasing the spores. But then spores like Sporobolomyces, which are actively shot off, often peak during the night when humidity is higher, aiding their discharge mechanism.
Wow. Altitude matters too.
Altitude matters. Studies clearly show far fewer spores at higher elevations, say above 2,000 meters, compared to down below 1,000 meters. The air gets thinner, literally.
Makes sense. And what about indoors versus outdoors? Do our homes offer a refuge?
Well, somewhat, but not entirely. Predictably, countryside areas tend to have higher overall spore counts than cities. Indoor air generally reflects the outdoor conditions, as air exchange happens.
But indoor sources can contribute too.
Absolutely. Damp rooms, mold growing on walls, even indoor plant material can create localized, sometimes very high, spore sources inside. And even just everyday activities, house cleaning, especially vacuuming, which stirs things up, or food preparation, are known to temporarily spike indoor spore counts.
So the invisible cloud follows us inside.
It does. And research continues to solidify these connections between specific spore types, concentrations, timing, and their impact on respiratory allergies and other health issues, making the invisible, well, tangible in its effects.
So we've journeyed from swimming spores navigating by chemical trails to fungi that literally launch themselves, or fire spore cannons, through the bizarre world of stinkhorns attracting flies, and finally to the microscopic, sometimes dangerous spores lurking in our own homes and the air we breathe. What really stands out to you from this whole deep dive into fungal spore dispersal and allergy?
Zooming out a bit, what's really striking is how the fungal kingdom's fundamental reliance on these tiny microscopic spores has driven this absolutely extraordinary array of evolutionary adaptations.
Just incredible diversity and strategy.
Exactly. From incredibly ingenious physical mechanisms like the actively shooting ascus and basidium we talked about, to really complex chemical lures designed to manipulate insects and even mammals into doing the dispersal work for them. These diverse and highly effective strategies are why fungi are truly ubiquitous. They're this silent, pervasive force literally shaping our environment.
But as we've seen, this ubiquity comes with a definite trade-off for us humans. While we've explored the incredible engineering behind their dispersal, we've also had to unpack the very real health implications.
Right. It's not all just fascinating biology.
No. From common allergies affecting millions to really serious occupational lung diseases like farmer's lung, and even touching on that contentious Stachybotrys scare, which really serves as a potent reminder, doesn't it? to always critically question correlation versus causation when we hear scientific claims, especially ones with big consequences.
That's such a crucial point. So maybe this raises an important question for you, the listener. Next time you take a breath, will you think differently about that invisible world swirling around you?
All those tiny travelers.
All those tiny travelers. Consider the incredible evolutionary pressures that shaped these fungal strategies over millions of years, and how something as small as a microscopic spore can have such an immense impact on global ecosystems, on agriculture, and directly on human health. Often without us ever seeing it happen, there's just always more to learn and discover in this hidden kingdom.
Well, we certainly hope this deep dive has given you a bit of a shortcut to being well-informed about fungal spores and maybe sparked some new aha moments about the truly intricate and amazing world of fungi. Thanks so much for joining us for this deep dive.